The following is the role of chemotaxis and microbial nutrition, brought to you by the Village Geniuses. So what is chemotaxis? Chemotaxis is the movement of an organism in response to a chemical stimulus, which may either be organic or inorganic. The chemical stimuli are called chemoattractants or chemorepellents, and either cause, as their name suggests, attraction to or repulsion from the stimuli respectively. An organism's motility is the ability to move spontaneously and actively, consuming energy in the process. Chemotaxis can be toward a high or low concentration. If the movement is toward a higher concentration, it is positive chemotaxis. If it is toward a lower concentration, then it is negative chemotaxis and is generally caused by a chemorepellent. Chemoattractants can be sugars, amino acids, organic sulfur, and inorganic nutrients such as nitrates, phosphate, and ammonia. Some examples of chemorepellents are alcohols and phenols, and extreme pH and oxygen concentrations. The nature of chemoattractants and chemorepellents can vary for different species of bacteria. One's chemoattractant can actually be another's chemorepellent. For example, phenol is an attractant for E. coli and a repellent for Salmonella typhimurium. Chemotaxis signaling in bacterial cells is achieved by the sensing of attractants or repellents by chemoreceptor proteins present on the outside surface of bacteria. The best studied examples of this are the methyl-accepting chemotaxis proteins, MCPs, and the Shea series proteins involved in the flagella movement in E. coli. The multiple MCPs found in either the periplasmic space or on the outer membrane are responsible for transmitting signals across the plasma membrane into the cytosol where they activate Shea proteins. Shea W and Shea A trigger autophosphorylation, which triggers Shea B and Shea Y to become activated. Shea Y then interacts with the flagella switch protein FLIM. This causes the flagella to assume clockwise rotation. Shea B, when activated, causes demethylation of the cytosolic side of the receptor, which promotes tumbling activity. When the levels of an attractant remain high, the level of phosphorylation of Shea A remains low. This also then means that the levels of activated Shea B and Shea Y remain low too. Thus, the flagella remain in a counterclockwise rotation, and the cell is able to assume a straight swim. The methylation of MCPs increases due to the absence of Shea B, but once the MCPs are fully methylated, they stop responding to attractants. The level of activated Shea A now starts to rise, and thus Shea B and Shea Y increases, and the cell now tumbles. With the presence of Shea B, the receptors are able to be demethylated again and respond to attractants once more, and the cycle can repeat itself if needed. In addition to traditional chemotaxis, there are subtypes, such as haptotaxis, where the gradient of a chemoattractant is expressed or bound on a surface, in contrast to the classical model of chemotaxis, in which the gradient develops in a soluble fluid, and chemokinesis, which is chemically prompted movement that, unlike chemotaxis, is usually randomly directed or not directional. There are multiple types of motility in bacteria, such as flagella motility, as discussed earlier. There's also amoeboid motility, which is a crawling-like motion usually seen in eukaryotes. There's gliding motility, which doesn't involve flagella. There's also pili or fimbriae motility. It is achieved by type 4 pili. They adhere to a surface and contract, pulling the bacteria forward. And swarming motility, which is the translocation of bacterial populations in a rapid and coordinated fashion. There is, of course, non-moving or stationary bacteria, such as Klebsiella pneumoniae, and Shigella, which have their food come to them. Necrotic cells can release chemoattractant molecules to which some bacteria are drawn. The release of these molecules triggers the movement of certain parasitic species. The same effect is found with apoptotic cells. Essential nutrients for microbial growth are either macronutrients, such as carbon, hydrogen, oxygen, sulfur, phosphorus, nitrogen, iron, potassium, calcium, and magnesium, or micronutrients such as manganese, nickel, zinc, copper, and cobalt. Because of the heterogeneous nature of the composition of the water sources that bacteria are normally found in, nutrients can be scarce. Chemotaxis towards organic nutrients is better characterized than towards inorganic nutrients. Some organic nutrients that have been observed to prompt aquatic chemotaxis are substrates such as sugars, amino acids, and organic sulfur compounds. Most bacterial populations use inorganic phosphorus as a phosphorus source, and in aquatic environments, this is a limiting factor to microbial growth. Chemotaxis towards inorganic phosphate has been shown to be exhibited in a number of bacterial species, particularly the Sphingobacteriales and also by a heterotrophic marine Thalassospira species. Bacteria needs to move to the highest concentration of nutrients. It does this with a type of primitive memory, where the receptors check to see if they are binding more or less of the compound than they were a moment ago. If they are binding more, the cell keeps swimming. But if they are binding less, it tumbles and swims in a random direction. The cell keeps randomly moving until a stimuli triggers a response again. The presence of nitrates, phosphates, or ammonia often trigger chemotactic movement. Chemotactic behavior may be a way for microbes to exploit microscopic changes in limited resources in the environment. Research has shown that ammonia elicits the strongest chemotaxic response to the majority of bacterial populations that were being tested from an isolated lake water sample. Ammonia elicits a chemotactic response for bacteria because they are one of the main components in amines, which are eventually incorporated in amino acids and proteins, which are essential for microbial growth. Some bacteria which have shown chemotactic responses to ammonia are the marine cyanobacterium species Cyanochococcus and a purple non-sulfur photosynthetic bacterium Rhodobacter spheroides. Nitrates have also been characterized as chemoattractants, with the examples of bacteria belonging to the family Chemotaxis, belongs to the order Sphingobacteriales. In a study conducted by Dennis et al., it was identified that some bacteria species belonging to the family Chemotaxis exhibited the strongest chemotaxis response to phosphate and nitrate. Some members of the Sphingobacteriales are capable of utilizing nitrate as a terminal electron acceptor in biochemical reactions, while some others aren't. It comes as no surprise that the members who utilized nitrate showed positive chemotaxis towards nitrate sources, enabling them to exploit an alternative electron acceptor source. On a larger scale, bacterial chemotaxis has been suggested as being important for the ecology and the maintenance overall of biochemical cycles of oceans. Bacteria play vital roles in their nutrient cycles, with the bulk of the biomass in the oceans being contributed to bacterial populations. In summary, chemotaxis can be a useful tool in determining microbial behavior in response to changing nutrient and/or toxic conditions. And with that, I'll leave you with a little history on chemotaxis. Back in 1703, Leeuwenhoek first observes motility. Almost 200 years later, Engelmann in 1881 and Pfeffer in 1884 make the first accurate recordings in bacterial movement. 25 years later, Jennings in 1906 records similar findings in ciliates. In 1908, our bearded friend Metchnikoff observes phagocytosis. 50 years later, chemotaxis assays were pioneered by Harris in 1953, and in more recent time, the discovery of a complement system by Adler in 1973.